In this tutorial, students, we are going to study about Morgan's experiments, and also we will understand the terms linkage and recombination. So by now we know that Thomas Hunt Morgan, he provided the experimental proof for the chromosomal theory of inheritance, and he worked with an organism called as the Drosophila melanogaster fly. Why he chose Drosophila for his experiments, also we know the reasons. So what did he do? He carried dihybrid crosses using this organism. So he carried several dihybrid crosses using these organisms, and mostly he wanted to study genes that are sex linked. So sex linked genes means the genes that are carried on the sex chromosomes, like the X chromosome. So, for example, one of the crosses here you can see is when he crossed a yellow bodied, white eyed female with a brown bodied, red eyed male. He got the progeny, and then what he did was he took the progeny also and he intercrossed the progeny, the F1 progeny. So, after conducting these experiments, he saw that these two genes, yellow body and white eye, did not segregate independently of each other, and the expected 9 is to 3 is to 3 is to 1 ratio was not uh, exactly 9 is to 3 is to 1, 3 is to 1, but it deviated slightly. So what I'm trying to say here is that after his experiments using these genes which were carried on the sex chromosomes, he found a slight deviation in this ratio, indicating that Mendel's rules were not completely followed in the case of these genes which are being carried on the same chromosome, that is your sex chromosome. So, after many such studies, they concluded that in case of genes, especially those present on the sex chromosome, if the two genes are sex linked, that is, they are situated on the same sex chromosome, okay, then they found that the parental combinations, that means exactly like the parent, the types exactly like the parents, are much higher than the non-parental types or the new recombinants. That means these genes are linked tightly to each other, therefore they will resist the recombinations and they will give you the parental combinations in higher percentages. So based on this, he coined two terms, linkage and recombination. So linkage means connection between genes or the physical association between genes on a chromosome, and recombination means when the genes are segregating and you are getting more of non-parental combination. So what is the connection between linkage and recombination? Like I told you, if there is a linkage between genes, the recombinations are less. Now, again, in linkage, there can be tight linkage and loose linkage. So, if the genes are tightly linked, okay, they are very tightly linked, then they will resist the urge to recombine, and therefore there will be less recombination. But if they are loosely linked genes on the same chromosome, then they will um, show more possibilities of recombining. This is to be kept in mind. Tight link, less recombination; loose linked, more recombinations. Okay, so that is this point. Uh, an example given here for you to understand linkage and recombination is, for example, if you take the genes for white and yellow, okay, so it will become more clear when we take the diagram. So we said that these genes white and yellow, white eyed and yellow bodied, are present on the sex chromosome, on the same X chromosome. So these genes white and yellow are tight linked, okay, or they have a tight linkage, because of which they will prefer to stay together and separate and assort together, and therefore you'll get more of the parental combination. So here you can see 98.7% are the parental type of flies. That means which have yellow bodies, white wings. Whereas recombinants are only few because these genes have preferred to stay together. So the recombination percentage 1.3%. Now, why is that? Because of tight linkage. And why the tight linkage or the tight connection? Because they are very close together on the chromosome. Whereas, look at this white wing and white wings and miniature wings. White colored wings, miniature means small wings. See how far the genes are located. So, when the location of the genes is further apart, the linkage is loose. So here it is tight linkage because they are close by, and here it is a loose linkage because they are far apart. So in this case, you can look at the recombinant frequency, it's much higher, 37.2%. Okay, so we go back to this slide, and here we see that same example wherein if the genes are further apart, then higher the recombination frequency, whereas if the genes are tightly linked, being close together, lower the recombination frequency, which we have seen in the example. So making use of this, it was Alfred Stratowind, who was again a student of Morgan, who used these frequencies of recombination to map genes. So what he said was that there is a distance between genes on chromosomes, and that can be easily told from the frequency of recombinations. If the genes are tightly linked, okay, then they will show lesser recombinations. If they are loosely linked, as we saw in this case, they will show higher recombinations. So he he kind of gave birth to the concept of gene maps, which are very very important in genetic studies today. So where exactly a gene is located on the chromosome is indicated in a gene map. Okay, so with this we finish what were Morgan's experiments. Then we have also looked at these two terms, linkage and recombination. Okay, so just to sum up, what is linkage? You have to know physical association of genes on a chromosome is called linkage. What is recombination? The generation of non-parental gene combinations is called recombination, and it occurs during meiosis. Okay, so you have to know these three things, that is Morgan's experiments and the terms linkage and recombination. And when is the recombination frequency higher? If the linkage between the genes is loose. But if it is a tighter linkage, then you have a less possibility of recombination.